Ernst, of course, is a leader in the area of the epigenome, and we look forward to your talk.
Okay, thank you, Alex, for giving me this opportunity to speak at the symposium. So this is a small portion of the human genome. In the previous session, we heard a lot about RNA-seq and sort of using it to explore a lot of the protein coding portions of the genome, but that's a very small percentage of the genome. And there's been a lot of interest in the sort of this vast non-protein coding portions and non-exonic portions because genome-wide association studies, sort of these unbiased assessments of where disease-associated genetic variations have been implicating sort of a large majority of the variation in these non-protein coding portions. So the question is, how do we start to sort of understand these portions of the genome and annotate them? And what I've done in some of my previous work is leveraging epigenetic marks, in particular maps of primarily of histone modifications mapped through a technology called ChIP-seq. And these are some of the core histone modifications, and they can mark different types of elements, enhancers, promoters, repressive elements, and so forth. But instead of looking at each mark in isolation, I had developed a method that would take multiple different epigenetic maps and, based on their combinatorial and spatial patterns, systematically annotate the genome in different types of states, what we termed chromatin states. So the underlying model for this is based on a multivariate hidden Markov model. So what we do is we assume that there's different classes of biological elements, such as enhancers, gene starts, and so forth, that we don't directly observe, and we don't need to know beforehand exactly what these types of elements are. What we do observe after pre-processing is at a 200 base pair resolution, binary presence or absence calls of different histone modifications. And what we do is we assume that those observations are coming from a hidden Markov model where in different states we have different probabilities of observing each modification. We model this with a product of independent Bernoulli random variables. So we learn all the parameters from the data. So in the first application of this methodology, what we did was we applied it to a large panel of histone modifications and related marks in a CD4 T cell data set. So the rows correspond to different states and the columns correspond to different input marks. So once we learn, and the darker values correspond to a greater probability of observing that modification in that state. And then once we learn one of these models, we can go across a genome and assign each location to being an instance of these states. So then, to give you an example of one of these states, this state was primarily associated with 3 histone modifications, H3K36ME3, K9ME3, and H4K20ME3, and had a very large enrichment for zinc finger genes and CAP1 binding. And this is, was described previously as a large portion of an abstract in a published paper, and then sort of showing lower values of these other marks. But what we were able to do here is rediscover this very specific biological concept by applying the appropriate computational machinery to the data, and then we also more systematically showed the lack of these other modifications associated with the state. And then with this work, I had gone through each of these states, overlapped it with a variety of other biological annotations and observation in terms of conserved elements, transcription factor binding, and gave different candidate annotations to the states, different classes of promoter, transcribed, enhancer elements, repressive, and repetitive elements. This was all in one cell type. One of the powers of the epigenomic data, unlike the DNA sequence, is that it can be dynamic across cell types. So there's variation with the different cell types and where the marks are. So this was in collaboration with Brad Bernstein's ENCODE production group. What we did was we took 9 of the subset of the histone modifications from before and mapped them across 9 human cell types. So this is looking at the data at one specific location. So each color set corresponds to a different cell type and then the different modifications in it. And what we did was we conceptually applied the same model as before, but we treated different cell types as if they were different chromosomes. So then the different so then we would get one common set of states across the different cell types, but we could have cell type specific state assignments. So the rows, again, correspond to different states. The columns correspond to different marks. And then we can overlap these states again with different types of other data, and then give candidate annotations. So this is another view of what we've effectively done, is we have the same genomic location in 4 different cell types. This is the raw input, and then we can summarize it into different chromatin state calls in terms of whether it's a candidate promoter, enhancer region, insulator, transcribe, repressive, and so forth. And then somebody can go into the genome browser and then quickly scan across those locations and see what the candidate annotations are. And I'm going to talk next about how to actually access this data. I worked in collaboration with the Roadmap Epigenomics Consortium, now applying this modeling approach across more than 100 cell and tissue types. My lab has had another number of collaborations applying this methodology to other data produced on campus and elsewhere, and many other groups are using either the annotations that are available or actually running the software on data they're producing. So I'm going to first, as sort of this is a sort of a tool-based talk about how one could actually access these chromatin state annotations. For a lot of applications, you don't necessarily need to run ChromHMM yourself. You might just be interested in obtaining the existing chromatin state model annotations. And then I'm going to talk briefly about how you could run the software on your own data that doesn't currently have chromatin state annotations. So this is the largest collection of epigenomic data with the chromatin state annotations. So this is showing you the grid of reference epigenomes produced by the Roadmap Epigenomics Consortium. So the rows correspond to different cell and tissue types grouped into sort of larger categories. And then the columns here correspond to different histone modifications primarily and a few other marks. And then blue means there is some observed data for that mark in that cell or tissue type. So in collaboration with the consortium, we had learned a model of based on 5 marks here that were mapped across all these reference epigenomes. And this is showing you the locations of the annotations at one genomic location. And then we had learned another model directly based on the observed data, also including H3K27 acetylation, that was defined based on the 98 reference genomes that also had this mark. But as you can see, this grid was incomplete, and if we kept on adding marks, we would reduce the number of cell types if we wanted to only run it on cell types for which we had every mark in every cell type. And if we didn't, it could confound downstream analyses. But I had developed an alternative strategy, which I won't have time to go into the method, but it's published, so you're welcome to sort of read it on your own. But the idea was that given we had this incomplete compendium, could we computationally fill in these missing experiments, leveraging the fact that we've seen other marks in that same cell or tissue type that we're trying to fill in the experiment for, and we've seen that mark in other cell and tissue types. So I leveraged sort of both types of information and then imputed every experiment here you're seeing. And then we had learned a chromatin state model based on the 12 marks that we were most confident in imputation based on being seen in enough other samples, and had defined a 25-state chromatin state model. So then this is showing you that annotation at one specific location. So all these 3 chromatin state models can be accessed through the UCSC Genome Browser custom track. And I'm going to walk you through that because it might not be obvious to everyone how to do that. And then this is just to summarize. We have these 3 models. These are the sort of color codings, the chromatin state annotations, and what were the original input marks that defined them. So if you want to load this up into the browser, it's defined on HG19 currently. So you would have the browser view on the UCSC. Then you would go to Track Hubs, and then you would want to connect to the Roadmap Epigenomics Integrative Analysis Hub. And to note that this is different than the Roadmap Epigenomics Data Complete Collection. They sound similar, but it's 2 different things. And then once you click Connect, it's going to take you back here to this main browser, and then you click Go to go back to where you were before. And then you now have this set of additional menu options. And then you would want to set this one to show, the first one here, and then click on it, and then you can go to ChromHMM, and then you can control which specific model you want displayed and the cell or tissue types. So right now, I have it checked that I want all of them imputed, and then you also need to sort of make sure the data type matches that, and I have it in dense mode. And then once you sort of submit that, then you can go back here, and now you have it loaded in the browser, the chromatin state annotation right there for all these cell and tissue types, and you can open other tracks you might be interested in and compare it. And now, briefly to talk about how you could go about running ChromHMM yourself. So you go to the ChromHMM website, you can download the software, and there's also a manual which gives you all the details. This is just a very quick thing to run it on the sample data. So you've downloaded it, you've unzipped it, assuming you have Java installed. Just open a command line, go into the ChromHMM directory, and then you can type in this command, which this specifies where the input data is, the output data, the number of states, and the assembly, and this controls the number of processors you're running. And then it'll output a model after a few minutes on the sample data. The actual input to ChromHMM, the modeling part is binarized data for this learn model command. To actually binarize the data, ChromHMM has commands that can do this based on aligned reads, either in BAM format or BED format, and that's described in the manual. So then after it runs, you're going to get a report like this, which at the top it summarizes what your parameters were, so you can remember that if you want to reproduce the analysis. Then you can see the emission parameters, the transition parameters, which I didn't mention before, but these are the probabilities between states. So the hidden Markov model also captures the spatial information through this. And then it has a segmentation file, which you can get in BED format, so it's giving you the coordinates and then what state it was assigned to. And then you can also have it in a format which is more browser-friendly, and you can load it into either the UCSC Genome browser or the IGV browser. And then it also has enrichments, so it has pre-computed or preloaded certain types of basic enrichments, such as where genes are, exons are, but you can also, either before you run it, add in additional BED files that you want enrichments computed for, or after you've run it, there's another command in ChromHMM called overlap enrichment, where you can take a previously learned segmentation and then compare it to additional data sets and create these types of heat maps. And you can also get positional plots showing you how states enrich relative to certain features, such as the start of genes. So to summarize this part, we have ChromHMM pre-computed across annotations across more than 100 cell and tissue types, and the software is available if you're interested in running it on your own data. And to acknowledge some of my collaborators for this work, and a lot of this was done in the context of either the ENCODE or Roadmap Epigenomics project. So, thanks.